I'm a recent assistant professor, but in preparing this talk, I thought about when I first met Martin, which was about a decade ago, which is kind of shocking. And I remember when I was assigned to him, he was my MPH advisor. I told him, I want nothing to do with the lab. And he said, why don't you volunteer and see how you feel about it? And 10 years later, I'm now an assistant professor here doing this work on the exposome. And that's really what drew me in and really fueled my passion and my interest in environmental health. And so it's an honor to really speak about this. And the final chapter of the Smith Lab, we know Martin's going to continue doing work and revolutionizing environmental health. But with the closing of the lab, I've inherited a lot of his legacy, literally and physically. And so I'm really appreciating the foundational structure that you provided as well. I think everyone's seen his foundational paper that he wrote with Professor Steve Rappaport on the exposome paradigm. And I think we know that Christopher Wilde introduced the idea in 2005, but it really had no traction. And it took about 5 years when this paper came out and really conceptualized how do we start thinking about the exposome and really think about how are we going to capture the totality of all exposures that occur throughout the life course. And this paper was seminal because it essentially formulated that we could measure internal and external exposures by measuring chemicals that were present in the blood. And so saying that you have to capture the internal environment, which could capture the inflammation and gut microbiome, which is critical in disease etiology, but also thinking about how the external environment becomes essentially embodied and causes biological activation of some chemicals, and then can also can contribute to disease etiology. And I remember I was most fascinated by this concept of stress. And I asked Martin, well, how are you going to really think about how that's embodied? And he said, as we heard, he likes a challenge. And he told me, that's your job. You figure that out. Go talk to people and come back and figure out a plan. And so that's really what's been motivating me in my career thus far. This paper also kind of really catapulted the field, and to date, there's over 1,400 publications that include the term exposome and PubMed, and I think we're going to continue to see exponential publications in this area, just showing how critical this paradigm is in the field. It's revolutionary. And so over the last 17 years since this idea was originally introduced, there's been advancements in the high resolution mass spectrometry and technology that extends beyond an analytical capacity to even identify what some of these well-known metabolites and these priority pollutants are. And so I think that analytical space is like an area that will continue to grow. There's been approaches to try and kind of resolve that with these meet in the middle approaches, using big, large population studies such as like Helix and exposomics, which are trying to match how the external chemical exposures, that the metabolites that map to those external exposures are then also linked to downstream health outcomes. And there's also network-based tools to try and understand how at multiple levels, from the proteomic to the metabolomic disruption, that chemicals can impact health. And so that's like, there's a lot of advancements in that space, and these are areas that I've identified that I would want to keep continuing to advance in the fields of exposomics moving forward. These are just 3 critical areas I want to highlight in my talk today, which are to remember the life course perspective, in that the exposome is the totality of exposures over the life course. And I think we need to start thinking about how to incorporate that into our studies. We've proposed how to use the exposome to assess cumulative risk. So I'll describe how we propose that, what I'm working towards, and hopefully hoping to advance. And also this field of addressing health disparities or health equity using the exposome. I think we haven't necessarily addressed that enough. And so thinking about that moving forward. And I also do want to highlight how Martin has contributed to advance. So the first one, incorporating time in the life course. So remembering that the exposome is dynamic. So every day we have different exposures of every day of your life. It's not static like the genome. And so we really need to capture that dynamic nature over time. Disease is also like the endpoint of a trajectory. And I think I like to think of we have exposures that occur over the lifetime, and that can really shape our trajectories over time. And our disease is really the endpoint of that trajectory. And that was proposed by in this paper by Paulo Vineas and Perucchi in 2022. And that remembering that the disease mechanism is multi-stage and really expresses this concept of the biography of a person. So over your life, you have all these different environmental exposures that really are shaped by the biography of the individual. And there's a concept in this paper that I really liked. And thank you, Martin, for still sending me papers to this day on topics that are relevant, where he proposes these, that we need to really think about the overarching determinants of these trajectories in life. And so one that they propose is this social economic position really shaping the intermediate exposures and their capital that influences health outcomes. So let me talk through this, where you have your constitutive biological capital where you're born with, and this can be influenced by intermediate exposures that change the biological capital, and that can influence health outcomes later in life. But we can't forget that these intermediate exposures are shaped by economic, social, and cultural capital. And so I'm really interested in how do we invest in these areas to help build the economic, social, and cultural capital of individuals to really shape and like change the trajectory of over the life course to make people more protected and more resilient over time. And then getting back to also these key stages to get snapshots of a person's exposome. How do we do that and what time? And Martin and Steve have been seminal in proposing these different periods of in utero, infancy, early childhood, adolescence, and adulthood as critical windows where there's a lot of biological change going on. And so one thing to note, a lot of these are during the early life because there's a lot of development. And so that's really shaped where my research focuses in early life, because I think that that's where interventions will be most critical to really shaping these trajectories over the life course. So next I'll move on to assessing cumulative risk. I think we've spent a lot of time during my graduate degree working on these ideas together with Cleona McHale, Dr. Cleona McHale and Dr. Sarah Daniels. And so this gets at the idea that we don't live in a vacuum, and traditional epidemiology research is looking at one exposure and disease outcome at a time. But in reality, we have, we're constantly exposed to multiple different pollutants, lived experiences, and there's heterogeneity within the population, right? That can influence whether someone may be at high risk versus low risk. And how do you really capture that? So we proposed in a commentary in EMM, which I'm grateful that Martin brought me in to really start developing some of these concepts and ideas, where we're really looking at if we want to look at how maybe the social stressors and the environmental exposures, the cumulative risk from both of those, we need to think about where do these things intersect. And so how do you measure this response or resilience? And so I've been really interested in this allostatic load concept, which is weathering or chronic wear and tear over time from stress, but thinking about do chemical exposures also influence these allostatic load biomarkers. And this is very preliminary data. It's just more proof of concept of the approach that I'm using in my research. So I'm working with the PEARL study, which is the pediatric ACEs and resilience study. It's based out of Benioff Children's Hospital, Oakland. It included 555 children at baseline, but it's a longitudinal study. but 2/3 of them were screened for adverse childhood experiences. And so I'm using that as my model to really understand how those adverse childhood experiences are biologically embedded over the life course. So this is a baseline. Children 1 to 11 years old. We have rich social determinant information. We have number of clinical outcomes and rich biological data, biospecimens. And so thinking about, okay, well, where do we start? We started by looking at the literature and looking at a number of allostatic load biomarkers to see if there's heterogeneity. A lot of times people make a composite score, but they don't really account for the fact that people vary in their response. And so I tried to do a cluster analysis, and I identified 3 clusters. I don't want to get too into the weeds of what that is, but when you look at health outcomes by the 3 clusters, this third cluster really has high prevalence of obesity and asthma. For example, the obesity prevalence is around 41%, which is around twofold the national average. And for asthma, national average is around 8, but in this subgroup, it's 64.4%. And so thinking about, well, this is an obese asthma phenotype in this cluster group. And this phenotype was interesting to me because it is a non-atopic, so it's non-allergic driven, and so it is not triggered by an allergen, and it's hard to treat. And so the question is, are there environmental contributions that may be explaining why this group has this phenotype? And so I'm working to use the rich social epi data that we have, along with potentially using an exposomic approach to really figure out why this cluster group has high allostatic load. And also there's one cluster here that seems to have high adversity exposure, but doesn't seem to have the same biological response. And so thinking about what may be protective factors that may be present in diet or lifestyle that may be explaining that resilience. And so I just want to end by discussing ways to address health disparity using the exposome. So the exposome has made a lot of advances in understanding like the biological complexity, but it hasn't really made as much progress with health equity, in my opinion. And so thinking about, okay, we're trying to characterize exposure with very detailed rigor. But are we doing the same thing with our social determinants of health? And so how do we really start to define those measurements with the same level of rigor to like get at what is the contribution of the social determinants of health to disease etiology? And then there's some models of like the public health exposome, the social exposome, and these concepts of using intersectionality that are trying to get at defining more of these structural components of the social environment, but there needs to be much more work in this area. And I think what's critical is also the recruitment retention of diverse study populations and researchers to make sure that who are we developing these technologies for, making sure that we really are capturing all, like representing all population members of the population. And so I just wanted to take a moment to also commend Martin for, I don't know if this is always intentional, but he always included, I think, very diverse populations. Even in his publications, we've recommended thinking about migration and thinking about these very nuanced social phenomenons and experiences. And Professor Sillier will discuss more about the populations that we've worked on as a group. And also, I think he really invested in supporting diverse trainees like myself. So thank you for all of that. And even in his last year, I was able to get him to help co-teach the EJ class, and through the Superfund Research Program, he did support the creation of an environmental justice class for the division. And so I think whenever called upon, Martin is always there. So I think that I just am grateful to have you as a mentor, and thank you for supporting my out of the box ideas. So with that, I'll just pass the baton to Fenna if she's available.